We were looking at the effects of deforestation on the diets of insectivorous bats. So we know that lots of bat species persist for the most part in forest that's been selectively logged, but we didn't really have much of an idea about their underlying ecology. So knowing that an animal is present doesn't really tell you that much about how they are likely to be present in future. So there may be time lags between logging occurring and the species slowly dying out. It may be that they're feeding on a low number of prey, and so they are then more vulnerable to environmental fluctuations. And so I was using DNA metabarcoding of bat guano to identify what bats have been consuming, and then creating big food webs essentially of bats and their prey to see how those differed between primary forest and areas that have been selectively logged. The main finding of interest really was that in logged forest sites, bats consumed a lower richness of prey than they were in primary forest areas. So the same bat species in an area that had never been touched with a chainsaw versus an area where lots of the best trees have been taken out, both those species of bat would persist, but in the logged areas they would be feeding on less prey species, so there were seemingly less prey available to them, either through the prey not being there or the habitat itself having changed so that the bats couldn't consume such a richness of prey themselves. And we also found that the networks had lower redundancy, so future environmental fluctuations could potentially harm the bats more in logged forest sites than those same fluctuations would in primary forest sites. So things such as climate change, El Nino events, etc. could have a greater effect on bats in areas that have been selectively logged. So we didn't really do anything with actually looking at the abundance of prey in the environment, but by looking at the data that I got from each bat's guano sample, we were able to pool the sequences we got into what we call operational taxonomic units. So essentially any sequences that are within a certain percentage of similarity to one another, we pool those together to be one unit. And so because there's not very good reference libraries of insect DNA for these areas of Southeast Asia, it's essentially just going, this is species A, species B, species C, species D, etc. And so it would be then, with that data, looking at the number of these operational taxonomic units per bat. So each bat would be, on average, consuming a lower number of these prey operational taxonomic units than they would be in primary rainforest. Whilst we know that logged forest is still much better than having no rainforest at all for the conservation of bats, it highlights that these forests can be vulnerable. So we need to be trying to protect them from future environmental perturbations and impacts wherever possible. So if you then go and degrade them further by human activities, that's likely to then have an impact on the bats that are occurring there, or we assume it is likely to. So it highlights that whilst these are valuable areas of refuge for the bats, ultimately they can be vulnerable, so we need to treat them with care. It's finished for now, yeah. I think there are lots of ways which we could try and build on this research, with maybe looking at larger areas of rainforest, more sites and things, but it's very intense getting the samples that we obtained, so a lot of it ultimately is very restricted by person power sort of that you can put into it, really. So it took nearly a year in the jungle in total for me to collect enough samples to be doing this, and so yeah, for us to be building on it again, it would require probably paying someone for many years to be sort of building up extra data for this sort of stuff. So for the time being, it's about as far as we're likely to go on this project, but there's lots of applications using DNA metabarcoding of predator diet to look at sort of ecology and ecological networks in future. So this was some of the first research to actually build networks of ecological interactions using metabarcoding data, which is the technology that we used. And so as we're streamlining these processes, we can probably start to look at worldwide a lot of sort of similar phenomena.